In this section, we are going to continue discussing translation, specifically the 3 nucleotide coding system. We're going to revisit our friends tRNA and rRNA, review the ribosomes and their role, and lastly talk about the process of creating proteins, as well as how we destroy these proteins when they have reached the end of their use. We will wrap up this topic by talking about how these processes contributed to the origins of life. Here are our lecture objectives for this section. As always, I expect you to have these mastered before our exam. Please let me know if you are struggling with any of these objectives. Here are the topics for Part 3 of the Chapter 7 lecture. Let's take a look at how tRNAs acquire their amino acid. There is a specific group of enzymes called aminoacetyl tRNA synthetases. In most organisms, there is one enzyme per amino acid. This means that the amino acid valine can only be attached to a specific valine-carrying tRNA via the valine aminoacetyl tRNA synthetase. To ensure that the right tRNA is present, the enzyme will check the anticodon as well as the sequences in the amino acid accepting arm. This serves as a proofreading step in the process, which helps ensure that the code in the mRNA sequence is correctly translated. The process of adding the amino acid to the tRNA uses the energy released by hydrolyzing ATP. This creates a high-energy bond that we will see come into play in a little bit. We've been dancing around the subject of ribosomes for quite some time. Let's take a moment to really get to know this organelle that is an enzyme. If you look back to Chapter 1, both prokaryotes and eukaryotes contain ribosomes. The eukaryotic ribosome is known as the 80S ribosome, while the prokaryote ribosome is known as the 70S. The structure of both is very similar. They both contain large and small subunits that are used to sandwich the RNA sequence. These subunits are made of rRNA and protein. Altogether, the ribosome is one of the largest and most complex enzymes we have. You may hear the term ribozyme used to describe the ribosome. When the scientific community discovered that some RNAs perform catalytic functions, we had to name them something to distinguish them from enzymes, which are protein catalysts, so we named RNA catalysts ribozymes. In the case of the ribosome, the rRNAs form the core structure of the enzyme, including the 3 docking stations for tRNA, where the protein polymerization process takes place. This makes the ribosome a ribozyme. Let's now take a look at the entire process of translation. This process begins with the initiator tRNA binding to the small subunit. This tRNA is always carrying a methionine group. When the start codon, which is always AUG, is read and recognized by the initiator tRNA, the large subunit will then bind. The large subunit will attach to the small subunit in such a way as to place the starting tRNA sequence in the P site of the large subunit. This begins the cycle of forming the primary structure of the protein. The cycle is a 4-step process and repeats as many times as needed based on the instructions in the mRNA. The first step in the cycle is the binding of the next needed tRNA to the mRNA sequence in the A site of the large subunit. The tRNA may only attach to the A site if its anticodon matches the mRNA codon in the A site. If it is a mismatch, it will be removed and the proper tRNA will be seeded. After the correct tRNA is in place, the second step is ready to occur. The aminoacetyl tRNA located in the P site will uncouple the carboxyl end of the end amino acid attached to it and form a covalent bond with the amino terminus of the amino acid attached to the tRNA in the A site. This process is facilitated by enzymatic regions in the large subunit. After this shift has occurred, the tRNA in the P site no longer has an amino acid attached, and the tRNA in the A site has the growing peptide chain bound to it. The third step occurs when the large subunit will shift over on the mRNA sequence, moving the tRNA in the P site to the E site and the tRNA in the A site to the P site. Following this, the fourth step rejoins the two subunits when the small subunit shifts over to match the large subunit. The tRNA in the E site will then exit and the process continues. The question then arises, how does it know when to stop? Just like initiation, there are specific sequences that tell the ribosome where the end of the gene is. We call them stop codons. However, instead of one termination sequence, we have 3 stop codons. These are UAA, UAG, and UGA. When these sequences are read by the ribosome, a protein known as the release factor will bind. This lack of subsequent tRNAs in the A site will release the amino acid sequence into the cytoplasm, and the ribosome subunits will dissociate from each other and from the mRNA. The free amino acid sequence will either fold itself correctly or may use a secondary protein known as a molecular chaperone to assist in the proper folding of the protein. The process is slightly different in prokaryotes, and if you are interested in that process, I will refer you to the textbook. While this process of translation can occur quite rapidly, sometimes it is still not fast enough when a large quantity of proteins are needed. In this case, the cell forms groups of ribosomes known as polyribosomes. These will form almost an assembly line, following one right after the other, to create the proteins needed by the cell. In bacteria, polyribosomes have been observed occurring even as the mRNA is being generated. In prokaryotes, this rapid creation of protein is possible because of the lack of mRNA processing and of a nuclear envelope. Protein synthesis is a mandatory system for all living cells. Given the differences between eukaryotic and prokaryotic systems, these differences serve as an excellent target for new antibiotics, since the drugs will, in theory, only attack the prokaryotic invader and not the eukaryotic host. Some of the best antibiotics have been isolated from other living organisms such as fungi. We have often found that natural products such as these work much better than synthetic drugs. Now that we have the proteins made, we need to address how the cell disposes of these proteins. Some proteins that are marked for disposal are those that have reached the end of their usefulness, while others are misfolded and must be destroyed before they can do damage to the cell. The cell utilizes a variety of mechanisms to recycle the amino acids in these proteins. All of the processes fall under the proteolytic pathway. One of these pathways is known as the ubiquitin proteasome system, or UPS for short. In this process, a series of enzymes work to tag proteins with a small protein called ubiquitin. This tagging indicates that the protein needs to be recycled. The actual recycling of the proteins are done by proteasomes. These enzymes are barrel-shaped, with the barrel surrounded by entrance and exit proteins. Inside the active site of the proteasome are proteases, which work to break down the proteins into amino acids for reuse. Let's spend a moment reviewing the entirety of the translation process in eukaryotes. We begin in the nucleus of the cell, where the RNA polymerase is attached to the Tata box initiator sequence by several transcription factors. These factors form the transcription initiation complex. As the RNA polymerase reads the gene, it creates a strand of RNA which is complementary to the DNA template. This RNA strand is specifically mRNA. Once the sequence has been finalized, the mRNA must be processed in the nucleus. The first process is the addition of the 5' cap made of guanine and a methyl group. The second is the addition of a poly A tail to the 3' end of the sequence. After these changes have occurred, the spliceosome removes the introns from the sequence by splicing the introns and sewing the exons together. The mature mRNA is then transferred from the nucleus to the cytoplasm. Once in the cytoplasm, the mRNA docks with the small subunit of the ribosome and an initiator tRNA. Once the start codon is found, the large subunit will bind to the complex. Along with tRNAs, the ribosome will build the polypeptide sequence until the stop codon is read. At this point, the release factor binds, dissociating the translation machine. The newly formed protein is then folded either on its own or by a chaperone into its correct structure. Sometimes the protein will be covalently modified to be activated or may be transported to the cell membrane. Before we close the book on this chapter, let's spend a little more time looking at the wonders of RNA. It has been argued that RNA pre-existed DNA in even modern cells. Researchers hypothesize that the presence of RNA is what helped facilitate life on Earth. As we have seen in this lecture, RNA has the ability to catalyze reactions. It can also store genetic information, as we have seen during the process of transcription and translation. This makes it a very powerful molecule for primitive cells. Let's explore these concepts a bit more. As we know from studying the laws of thermodynamics, for life to function, it must have the ability to catalyze reactions. This is because most reactions will not occur on their own, and even if they did, they could take a very, very long time. In looking at the macromolecules in our cells, only 2 macromolecules have this ability: nucleic acids and proteins. While proteins are very catalytic, no enzymatic reactions allow proteins to reproduce themselves. Only RNA has this ability. These 2 properties are starting to look a lot like the properties of life we are always talking about in biology, and so far, the only macromolecule capable of demonstrating these traits is RNA. Besides reproducing itself, another important aspect of life is the ability to pass on genetic information. As we have learned, replication needs enzymes to help the process occur. But in a world without these enzymes, RNA is able to continuously copy its sequence. We can see a hint of this in the RNA primase used in DNA replication. This way, the cell has the ability to pass genetic information to the next generation with minimal machinery. We can even see this ancient way of passing genetic information echoed in some present-day viruses that have RNA genomes. While viruses are classified as half-living due to their reliance on a host cell and their lack of important cellular components, we can see how the beginning of life may truly reside with the RNA sequence. All these things lead to the hypothesis that RNA predates DNA in the evolutionary pathway. RNA could have served as the perfect vehicle at the dawn of life. It allows for the properties of life to exist. Only when things start getting more complicated in the early prokaryotes could a more stable system, such as DNA with its modified sugar and base, be needed to sustain life. Not bad for a molecule we don't talk about all that often. Well, that's it for this chapter. Here's a summary of the things that we've covered in the 3 parts of the lecture. Let me know if you have any questions.